Hello, Connor.
Hi, Fergus. How's it going?
Yeah, good. Yourself?
Yeah. Good. Yeah.
Dear Nicola, should we start?
Yes, sure, we can start. I think Connor's just stepped away for a second, but he's here. That's great. Okay, I think we are now live and ready to begin. So hello and welcome everyone, especially good afternoon to those of you who are joining us from Kazakhstan, and good morning to those of you who are joining us from the UK. It gives me great pleasure to welcome you to this third webinar in the Research and Enterprise webinar series. My name is Nicola Darling from the University of Dundee, and I will be moderating this session. So the theme for today, today's researchers, is the emerging technologies in healthcare. The format will be a discussion, largely directed by the questions which you submit to us. So if you're joining us on Zoom, please do submit your questions via the chat box. And if you're joining us on Facebook, you can also submit your questions there too. So today I'm joined by researchers from the School of Life Sciences at the University of Dundee. So Fergus and Connor are with us. So perhaps you could introduce yourselves. Maybe Fergus, could you begin?
Yeah. So yeah, thank you, and good afternoon, good morning, everyone. So I mean, history of me. So I did an undergrad in chemistry, and then I wanted to specialize a bit more. So I then did a master's in chemistry, and then worked in industry for a year. And then I took up this PhD. So I'm now a year and a half in. So I'm in my second year, working in chemistry, more drug discovery based within the drug discovery unit in School of Life Sciences.
Great, thank you, Fergus. Connor.
Yep. So I am currently a third year PhD student at Dundee University, just early third year, and I started my career at Dundee as well. I did my undergraduate here in microbiology, and then expanded into doing cancer research for my master's at Nine Wells University, which is an extension of our campus. And then, yeah, I started my PhD, and then I work for Professor Julian and Professor Alessio Ciulli here at Dundee Uni.
Great, thank you, Connor. And so I'm interested to start with, back at the very beginning, why did you decide to do a PhD? Fergus.
That elusive question. So I finished my master's. I didn't really know what I wanted to do. If I committed to a PhD, it would be three and a half years of dedication to a field that I don't necessarily at the time think I wanted to go into. So I applied to some places, got rejected. That is also worth noting. You might get rejected from your first choice. So I went into industry for a year just to make sure I actually liked what I wanted to do, which was chemistry. And then about a year in, I decided I wanted to do a PhD just because I wanted more freedom to do my own research rather than being told by a company boss to just make this compound because we want you to, and you're just an individual rather than your own independent investigator or a researcher. And then I think having a PhD within science is always a good thing to have. People say within an industry there's kind of a bit of a glass ceiling if you don't have one, so you can only really get as high as you want to get without one. At least that's true for chemistry. I'm not going to generalize for other disciplines. So yeah, it was both wanted to do it and also for future career aspirations, it's better to have one.
Sure, that makes sense. So despite being rejected initially when you applied for PhDs, you continue to persist and try and follow the idea of doing a PhD anyway.
Yeah, that's
which is great. I think we've all been through that stage of applying for things and being turned down, unfortunately.
Yeah,
that's a useful message. Connor.
Yeah, so after my undergraduate, I was kind of in the same boat as Fergus. I didn't really know if research was going to be for me, because in your undergraduate, the way it's structured is you tend to only get things like 8 to 10 weeks in the lab. It's really your first major experience in a lab. But I chose to do a research master's. So I spent an extended time, I think it was about 9 months in a lab setting. And I kind of fell in love with the sort of the structure of doing lab-based research, at least from an academic perspective. And from there, I just sort of decided that for the very moment in my life, I thought I would like to continue doing that kind of process of being able to make my own decisions in the lab and being able to do some sort of problem solving, which although I don't have any industrial experience, I get the impression that it's a lot more structured based, a lot more focused on the goal. Whereas I think I like the idea of being able to sort of explore around an area and make mistakes and sort of, trial and error type stuff. And that's kind of where I came from with that. And yeah, so I chose to do or apply for a PhD, and then Dundee asked for an interview, and I got in a week later. So yeah.
That's great. And a speedy response there from Dundee.
Well, it was quite funny actually. I was actually in Germany when I got the interview. They asked for an interview, and it was the day I came back from Germany from a holiday. And then I had the interview, and then yeah, I was just happened to be lucky that I was asked to. I got given a letter the next week, and then that was it. So I was kind of lucky because it's the only one I applied for. So, but yeah.
Well, that's brilliant. And Fergus, I'm interested, your PhD has a partnership with an industrial partner, Benalis, is it?
Yeah.
So was that something that you were really looking for when you were applying for your PhD, an industry partnership in the PhD?
Yeah, so when I first applied, we supervisor Paul Wyatt hadn't got the industry on board yet. So my PhD is funded by the Medical Research Council. But they wanted to make it an I case, so an industrial case PhD, where you partner with your industry, and then bonuses, you get paid a bit more, and you have the interactions with the company. But I thought, almost coming from an industry and then also having an industry as part of my PhD, it kind of, it would improve my learning, I'd hope. And so whilst I'm doing my research here, I also sit in on some meetings that they have where they're based within a drug discovery company rather than the research institute that we are. And then part of it as well, I have to spend 3 months working for them. So I saw that as, if I work for them for 3 months, really like it, really impress them, I can finish my PhD and then hopefully just springboard into a job, or at least a strong application to go work for them. So it's definitely something that enticed me as well.
And it sounds like your interaction with this company has had a big influence on your PhD as well,
Yes.
because of your ability to experience how the company is run.
And we have quite a good open dialogue as well. So there's not much confidentiality that we keep to ourselves. So what I make, I share with them, and kind of vice versa. But it's very open, which is always appreciated, because obviously there's some industry and academia secrets that you don't want to share. But it's quite good. If I have an issue, I can show them my chemistry, show them my problems, and they will come back to me in a confidential manner. Obviously, I'm not going to share it, but no, it's just very good and quick replies as well, which is always good from industry.
That's great. And you also had in mind what you might want to do after your PhD.
Yeah, so probably at the moment, postdocing it's not really appealing to me just yet. It might do come on my final years, but no, at the moment it is going into industry and then working within the drug discovery industry, getting the experience there, and then hopefully improving my skill set and knowledge of the company and industry as a whole.
That's great. And Connor, have you thought about what you might want to do after your PhD?
Yeah, I mean, obviously my goalposts are a little bit closer in that sense. I'm going to be graduating, well, we'll see, but I mean, I think the goal is to hand in my thesis next year. So yeah, I've been thinking a little bit about it. I figure at some point I'd like to try industry. It's definitely on my list of things I would like to sort of see the difference. But realistically, I think what's probably going to happen to begin with is to do some sort of postdoc at some university somewhere. Ideally, I'd like to move. I think that's something I definitely is on my list. Once I graduate is to leave Dundee for the first time in 10 years. So that would be yeah, that's definitely there. But yeah, I'm not sure yet, and I haven't got really any major plans just yet. So
is postdocing what you want to do, or you think you're kind of driven into?
So I think so maybe it's probably from a biology perspective, I think I need to have a little bit more experience in different areas. I think that would help me for the industrial case. Ideally, I think realistically, I don't want to be an academic forever, and I certainly don't want to go down the PI route. It's not. I don't think I could handle that stress, but I think I don't know. There is definitely an appeal to industry, but I think as a biologist, I need to sort of expand my toolkit or my what I'm good at, so that way I can be more appealing. So I think doing a postdoc for 4 years isn't really going to harm me too much, I don't think.
No.
So, but even if it's not 4 years, I know there's smaller ones. I know there's even 6-month placements I can do. So, but yeah, I think industry long term, but short term, I think just to get the experience, I'm happy to go for lower pay and harder, more stress. I'd argue maybe not more stressful, but more. You know what I mean? Like with a PI and stuff, it's probably more like 1-to-1. You know, I want to do that for a bit so I can get better at my craft. So yeah, that's my thought anyway.
That's great, thank you. And I think Connor, you did rotations before you started your PhD.
Yeah.
Could you talk a little bit about that? Yeah,
so the way it's structured at Dundee is that we have, I believe it's either 2 or 3 different programs. I think both of them are funded either by the Mrc or the Wellcome Trust in Britain. But these are 2 or 3 PhD programs where the first year of a 4-year PhD is done in different labs, where the labs are about 3 months each in length, where you're given the opportunity not only to experience what that lab does, but you're also sort of given some of the training tools you need in terms of writing. You learn how to write sort of in an academic sense, and you have to present like a miniature viva every single 3 months. And the reason why I chose to do that was, coming out of my master's, I wasn't actually 100% sure where I wanted to go. I had a sort of a rough idea, but I thought, getting those extra 3 months in to do things I might not get to do. For example, when I did my rotations, I effectively chose 3 disparate labs, and one of the labs I chose that I ended up not going with was an animal lab, a lab that worked with mice. So I got the opportunity to experience working in an animal house and doing that sort of aspect of biology. And one of the other benefits I got from this, in particular to my project, is that the other 2 labs that I actually chose both of them, and ended up making my own new project that was completely the brainchild of a couple of discussions we had between my main PI and my secondary PI. And yeah, if I hadn't gone down that route, I wouldn't have actually had any opportunity. This wasn't like a set predetermined project. This was a project I had to wrangle myself. And so from that, I've been able to do a completely unique project that I wouldn't have done otherwise. So yeah, it's a really, really useful tool, I think, for some people who maybe have come out of their masters and not 100% sure what they want to do in terms of, I mean, if you're a biologist, you can specialize in almost anything. And I think it's good to be able to, maybe try some microbiology or try some cancer research or looking at DNA replication, and just seeing what the different techniques and what you can expect from working for 3 years of your life on something that you're going to be very intimate with will actually be like. And so yeah, that's I would highly recommend it to anyone, is to do a rotation program. I really enjoyed it. It's a good year to get set in as well. It's almost like the shallow end of the pool type thing, because although it's at a PhD level, it's like a miniature PhD project you have to do in each one, and obviously it's not got quite the same stress as doing a thesis. So but I really enjoyed it. Yeah, it was a very useful aspect to my PhD. So
yeah, it sounds like you've really benefited from having the rotations in terms of developing your own project idea as a result of it. Which is,
yeah, I think that was just very lucky, but yeah, no, definitely. It was, it was just good to, I mean, obviously working for Alessio, what he does is very generalizable to a lot of different other fields. But yeah, I think I was a combination of luck and also, just, coming up with an idea and then broaching it with the PIs, and they're always up for doing new wacky experiments. So that's I really enjoyed it. It was a great first year as well. So I think it's also really useful because I mean, there are, I guess, if you start your PhD and you find you actually don't want to do that kind of stuff, if it turns out that either you don't like the sort of the way it's structured, or you don't like the stress, or you don't like actually doing lab research, I think this is doing a rotation year is also very useful, because I think it sort of sets the tone of what a PhD is in a slightly more relaxed setting and a more understanding setting. There is a lot of we have a lot of one-to-ones with the heads of our PhD grant program at the time, and if you're feeling like you don't want to continue, I think it's probably a much easier a way to just say, look, I would like to phase out, rather than having to face your PI, I guess, once you're in a project. And they'll also tell you whether or not they think that there's any issues going forward or what you need to improve upon. So yeah, I think it's a really useful way of doing a PhD. Although I know it's not for everyone. I know some people are dedicated towards working on one project, but I just felt that I didn't really know I knew kind of roughly what I wanted, but I didn't necessarily think that what I wanted in that moment necessarily was going to be reflective on what I truly wanted later on, because I didn't really know enough. And honestly, I think it's, I'm willing to say I didn't actually think I knew enough biology to sort of know every single field. So it was definitely helpful in that sense.
That's great. Yeah, it's quite a big change when you move from undergraduate to doing a PhD and doing research. It's quite a difference. So
massive,
the rotations can help, I guess, with the sort of transitioning into that new research field. And Fergus, did you do rotations as part of your PhD, or how did you apply for yours?
No, so I'm the complete opposite to Connor. I jumped straight in the deep end. So it was quite hard to transition because when I worked for a year in industry, we were large scale production. So working on the multi-kilograms for fine chemicals in the pharmaceutical industry. So going from a year of where your mindset is large scale to jump into research where you're really only working on milligram sort of reactions it took, say, a couple of months just to kind of get used to it, as well as get used to, this is my PhD, I have to determine the outcomes, what I want from it. And it was much more independent thinking than rather, as I mentioned earlier, being told what to do because you have to do it. So yeah, it was a steep learning curve. So as Connor said, we have a thesis committee, so we meet with them every 6 months just to make sure that your goals are on track with what they also think as well. It's like they almost look to see whether you are compatible and if your research has legs, so to speak. So do they think it can continue for the full term of your PhD? But no, once I got over that initial hurdle, it's been pretty smooth sailing, so to speak. Obviously, COVID has caused a major disruption to everything. But no, it's good now. I probably, looking back, yeah, maybe a rotation probably would have been better. But because I'm from training, I'm a chemist, and within the School of Bioscience, which is predominantly more biology and biology focused, I think to do a chemistry rotation in that would be a bit more challenging than it is necessarily for biology. But then it's not necessarily a bad thing. It's always good in your early 20s to learn what you like and learn what you don't like. So yeah, probably if I were to change anything, it might have been to do a rotation, but I don't have many regrets.
That's good. I'm glad to hear that. And you kind of touched on the COVID-19 situation. Perhaps you could share how that has impacted your research and what you've been doing since the lockdown situation started.
Yeah, so we've now been in lockdown for 14 weeks, which, looking back, it's gone incredibly quickly and incredibly slowly all at the same time. It kind of came at a bit of a, not a bad time, it came at a bad time for everyone, but just as my chemistry and started to work, so we were told to close the lab down. So it's taken a massive step back from lab-based and more. Within this 14 weeks, I've just been writing up my thesis. So I've got all my introduction done, my discussion, my chemistry, my experimental. So that, if you were to look at a PhD timeline, it's kind of what you do towards the latter year, the latter 6 months of your PhD is just writing up. I mean, I've been lucky enough that I've been able to do this now. But I've kind of come to a bit of a bit of a lockerhead with what I can do, because I've done all I can do from home. And obviously a PhD in science is always experimental. So there's only so much you can do without being in the lab. So I've spoken with MRC. So will be applying for an extension towards the end of my PhD. And that is simply because having 3 months out of a lab, out of a 3.5-year PhD program, is a hefty chunk of that. When second and third years, they like to say is the crucial years where the chemistry works and the science works and you get your results. So touch wood, I hope I don't end up using it, but it's always good just to have it as a safety net in case I will need it. So yeah, that's kind of it. COVID is a nightmare.
Yeah, it's a bit of a challenging situation, particularly when it's a research-based, experimental-based PhD that you're doing.
Yeah, exactly.
It sounds like you've been using the time well, though, working on your thesis.
Yeah.
And Connor, can you comment on how it's impacted your research?
Yeah, so with Fergus, what he said, essentially the same, but I would just say that instead of not needing it, I'm taking it. If I can get an extension, I will be taking it. But yeah, I mean, so what have I been doing? Essentially, obviously, being that I'm a little ahead of Fergus in terms of the timeline, I've been doing essentially the same thing. And about 3 weeks ago, I went to standstill. I've done as much as I can outside of the lab. But yeah, I mean, at the same time, I have had to do some writing as well. We published a book chapter, Alessio's group, during this time. So I had to do that for the first few weeks. But yeah, unfortunately, with the COVID-19, and much like what Fergus said, I think March was just a really successful month for everybody in the research facility, because I've found that a lot of people were very successful just before lockdown. And in my case, I actually had a successful CRISPR clones that I now had to bin, essentially, just going into April. So yeah, I mean, I've lost not only the 14 weeks out of the lab, but I think I lost probably about a month's worth of work before that, at least because through trial and error. So, and how many more weeks I'm going to lose restarting the lab over the next few weeks. But yeah, I mean, it's not been an ideal situation. I think we'll probably be okay in terms of getting back into the lab, and I think most of the grant funders will be quite reasonable and give us something, especially I think those that might be graduating in the next year. So fingers crossed anyway. I hope so, or else I'm going to be quite short in terms of my research goals before the end of my PhD. But yeah, short of that, it's just been, you try and do as much as you can, but there's only so much you can do in an office chair sitting in your house. So, but yeah, just make the best of the time you have, despite the circumstances. And hopefully we'll get back into the lab soon and get everything up and running and back to full force within the month. But we'll see.
Fingers crossed.
Fingers crossed.
I hope you manage to get everything back up and running very quickly after we get back.
Sure.
I was wondering if you've had support from the university or felt supported by the university during the COVID lockdown. Connor?
So in terms of our help and assistance, we've had quite a few services that have come out and helped us if need be. Personally, I haven't really taken any of the help. I don't think I need it personally, but we've had, for instance, we've had mental health, and we have our own, as far as I understand it, dedicated guidance type person, HR person for all the PhD students at the research facility. And from what I understand, there is assistance on that behalf. In terms of more just the structure of the PhD itself, yeah, I've been in one-to-one contact with my PI more or less every week over the 14 weeks. Which is more than I would say normally during the research time when he's traveling. So yeah, I've had a lot of one-to-one assistance, and yeah, there's just been a lot of updates every day on tracks and getting back, and taking time off, and encouraging us to take a personal day here and there, because it can be quite stressful working from home with all the distractions and uncomfortable office chairs and stuff. But yeah, we've had, in my opinion, I think we've had sufficient and ample resources available to us for during this pandemic. So
Mm-hmm.
yeah,
it's really good. Fergus, have you felt supported over the lockdown period?
Yeah, so just kind of agreeing with Connor. So the PhD office sent us like these 5 questions every month, every other month. It was like, are you getting enough supervision from your supervisor? Do you feel like you're suffering with mental health issues, or do you think, have you seen a decline in your productivity? Which I think is largely yes. And yeah, as Connor said, there's like, if you say yes to any of the productivity or mental health issues, they are pretty quick to say, here's our guidance counsellor, here are some useful links just to have a read about. And then as Connor said as well, it's like Connor's PI Alessio and my Paul Wyatt, they are extremely busy, important people within the school. So they are always travelling, they're always in meetings. But it has been, we've moved from once a fortnight or once every 3 weeks to once a week updates, just so we kind of have an open dialogue as to where we are. Like, I've openly told Paul that my productivity has slumped to about 50%. Working from home, I'm only really doing about 4 hours of work a day. That was until last week or so, when I'm pretty much at a standstill with what I can do. And he's okay with that. They expected it. They expected productivity to decline, obviously, because scientists, it's quite hard to do work from home. So they've been pretty supportive of that. Or if I've said I have not a lot I can do, they have probed me in like, well, have you considered doing this, like bringing this into your introduction of your thesis? Or, well, if you've got this time, why don't you look at something you necessarily wouldn't consider? So yeah, I think as PI and both as a school, they have been really quite helpful.
That's really good to hear, that you've been feeling supported by the university and by your supervisor, especially during this time. So Yes, let me just have a quick check through my questions here. Well, I was wondering if we can talk a little bit about your research. I have a couple of questions in about your research. So, Fergus, your research is using fragment-based screening?
Yep.
That's right, okay. So there's questions about how you do the screening and how you optimize the screens. Maybe you could talk a little bit about the screening process that you use?
So it's a good question. At the moment, I don't do any of the screening. That's mainly because I haven't got to a point where I have compounds that I can screen. So, I mean, as a whole, I'm sure hopefully Connor might be able to help me out. From the School of Life Science, we have fragment-based drug discovery, you have X-ray crystallography, where you can co-crystallize your fragments, so your small molecule, into a protein, and then you can see the binding pockets and the binding interactions that it makes. What else do we have, Connor? We have NMR, so nuclear magnetic resonance. And so again, you put your compound into a solution with a protein, and then as it binds towards the protein, you see a sharp increase in the signal of the binding and protein, the compound and protein binding. And so in the free state, you'll see, for example, a peak here that correlates to the compound and a peak here that correlates to the protein. But as they come together, you'll then see a peak. So you shouldn't be able to see any more protein, you shouldn't be able to see any more compound, because they should be completely bound and optimized that way. In terms of optimization, I don't necessarily know, because I haven't had to experience that yet. I don't know.
The ITC, SPR, those kind of techniques, right?
So
once you find a compound, you then do. I mean, at least in our lab, it's ITC is the first thing, right? So you, well, I mean, I'm not technically a biophysicist, but I mean, I've done ITC. So ITC is essentially you put your compound, you either put your compound in a solution that is calibrated to a reference cell, and then you put your ligand that you think it interacts with, sorry, the ligand and the protein of interest, and there's a change in temperature that can occur, and that's measured, and obviously the more rapid change in temperature is indicative of an increased affinity. And then SPR, although I don't understand how that fully works, it's basically something to do with gold landing on the surface.
So you put your protein on a gold plate, and then you shine light at the plate. But as you're doing that, you're then also titrating in your compound. So then, say for example, your compound binds with the protein, the refractive index, so the bounce back that the light gives, should be then different. Your refractive index of a protein bound to the compound should be different than the refractive index of the free protein.
Yeah. And there's a few other approaches, right? Yeah, but I mean, once you've got a compound, that's the bit where you start messing about with it, from what I understand.
Yeah, but yeah, as Connor said, that's more biophysics, and we have postdocs who specialize just in that.
Yeah.
Yeah, this is quite a big area, trying to optimize and complete all of the screens depending on what your target is, I guess, as well.
Yeah, but very.
You don't have to have the structure of your target protein in order to do this kind of screen, or you do need your structure?
I don't know.
Well, isn't there there's two different ways, isn't it? Reverse and forward, right? It's like whether or not you're probing from, you're probing a target that you don't, or you're probing the phenotype of a cell, or you're probing, if you know the structure, then you do it the other way. So you make compounds the other way around, I think, right? That's the, but
so I think, you can do different experiments if you know the protein compared to if you're doing a generic one against a load of proteins.
Yeah.
But as for that, I haven't been involved in that yet. I haven't got to the point where I can speak confidently about the process.
Fair enough. Yeah, it's you're just before the stage, really, I guess, for doing this. But I don't know whether you can comment on whether you use AI in order to design the fragments and optimize the fragments.
That's a very good question. So I attended just before COVID in March an AI conference on chemical reaction outcome predictions. So that was looking at how can you use artificial intelligence and machine learning to predict a chemistry reaction. So a lot of time it's kind of trial and error, or a chemist will know that reaction won't work. But then they're looking at how can we teach AI to know that that reaction will work rather than wasting time in the lab. As for me, yes, I have used AI for these fragments. So I've applied what we call a synthetic accessibility score. So this is an artificial intelligence measure that the computer takes into consideration how complex is the compound that you're looking at. And then again, it looks at how complex it thinks the chemistry is to synthesize that compound. Some of the results are a bit obscure and a bit iffy, mainly because it's still in production, so to speak. It's still being made. But yeah, that has definitely guided us a lot, rather than just looking at these fragments and going, well, I want to make that one, just because I like the look of it, rather than what the chemistry and the AI in this case is telling me to look at.
Yes. Great. It sounds like it's got a lot of promise in that area, although perhaps early stages, again, for developing AI.
AI is up and coming. You know, a lot of software is available where you can put your target molecule in and it will break it down into a much simpler molecule, and then also provide you with a forward route to that molecule. So that area is not necessarily new. So these programs have been around. So SciFinder and Reaxis, these programs have been around for about a couple of decades now, but it's only now that companies are actually really realizing their necessity for chemistry and to streamline the chemical process. So there are a lot more programs coming out, as well as the current existing ones have developed a whole lot more what they're telling the programs can and cannot do. So yeah, it's emerging, and you better get learning it quickly.
It sounds like there's lots for you to work on in that area then, if that's something that you're interested in.
Yeah.
And Connor, perhaps I could ask you a couple of questions about your research as well.
Yeah, no problem.
So you discussed there's a proportion of the proteome which is undruggable.
Yep.
What proportion? Do you know? Is it a large proportion?
So I can't remember the exact number from, but I think there was a paper in 2011, if I'm not mistaken. There is at least 20% of the total proteome that is undruggable to some extent. Yeah, I think it's maybe it's 20, or maybe it's maybe it's 80 actually. It's one or the other. I can't remember which way that flips, but yeah, there's a certain proportion, and I think it's mostly down to a combination of things. Either there's not enough sufficient binding pockets. I mean, it really depends on obviously what you're targeting. If you're targeting not only the protein or a group of proteins, whether or not you're targeting a surface that can be that can actually be targeted based off of its structural alignment, and obviously if you're using an inhibitor, you know, there's a lot of, I mean, there has been in the past a lot of inhibitors based off of ATP, as in like the ATP binding pocket. But the problem is that if you target a similar a surface area that it has a lot of similarity with a lot of other proteins, because a lot of proteins have ATP binding pockets, then you're going to have serious problems, because in order for you to get the desired effect, you're going to have a high level of your inhibitor inside the cell, and that's going to lead to serious off-target effects. You know, if it's targeting one ATP, there's guarantee there'll be one that's similar enough that it's going to also have a negative impact on it. So yeah, there is definitely
Sorry, Connor, for the undruggability, is that something it's generally known, or has your lab or you had to find that out for yourself?
No, it's been written about in the past. There is just there is a recognition that there is just some proteins that that seem to have none of the characteristics necessary to make them good targets for being druggable.
Okay.
I can't remember what the paper is. It was in Nature. Yeah, it's also one of the early arguments made for the development of PROTACs was the fact that there are essentially proteins out there that you can't really target using an inhibitor, and whereas we could with a PROTAC, because you're not necessarily, well, you're not using an occupancy-based model, you're using a degron essentially, or a targeted protein degradation approach instead. And that's one of the benefits of these new technologies like molecular glues and PROTACs, for example. So yeah.
And then for the Bromo-Tag that you were discussing in your presentation, you'd be adding a tag onto your protein to then target it for degradation.
Yeah.
How quickly can your target protein be degraded using this?
So using our most recent PROTAC that we've developed, my half-life for one of my targets that I've CRISPRed into, it is 24 minutes. 24 minutes to get half maximal degradation. So within an hour, we can get essentially almost all the protein degraded, at least for the target I have. Obviously, I haven't done enough tests yet to say whether or not this is applicable to all proteins, because I guess the kinetics will be different for proteins that might be in different localizations within the cell, or maybe are in some sort of scaffolding or something. These might be a problem, or if you're using membrane proteins, I don't know yet. Maybe we may not even be able to target membrane proteins just because they tend to be dealt with using a different system. But yeah, based off of the experiments I've done so far, we have been able to get selective and potent degradation within 24 minutes, or at least the half maximal degradation within 24 minutes, which is it's fast, and comparing it to a lot of the other degron technologies, it's well within the reasonable range. As far as I understand, Auxin is as fast, but I mean, you can also make the point that each one of these tools has their own positives and negatives, and with Auxin, you also have to have in the sort of the scaffolding protein involved as well, which means you have to have another factor added in that you wouldn't have to have with my system, or the system I'm working on rather. But yeah.
So that is pretty fast to get degradation of your target protein. Wow, it's amazing.
It's yeah. I mean, it was a surprise to us. I mean, we've went through 8 renditions of 8 different compound iterations. So, and the thing is, even last year we didn't have a single selective compound. So to get from essentially nothing to 24 minutes in a year is not bad. So I'm very happy with that.
Yeah, that's amazing. So we have a question in. Both of you are looking at targeting proteins, perhaps to look for therapeutic benefit. Could you talk about what happens next after you've discovered something and you've shown that it's got therapeutic benefit? What happens next, and how long does it take to show that it is a suitable drug that could be used in the clinic, for example?
Wow. I mean,
Fergus, you first.
Unless you want to, I can talk from the chemistry, and maybe you can talk from biology.
Sure.
So from a chemist perspective, once a protein has been identified, you will then more than likely use computational methods to really access and see what the space is within that protein structure. So how much of it, where in that structure could you put a molecule, and what is the relative space that you're trying to probe? So once you've then done that, so from there you'll from the computational aspect of it, you'll look to see, here are the amino acids that they're interacting with, or here's a water pocket, for example, that you could form an interaction with. You'll then go and design some compounds based on what your computational modeling has shown. And it's also incredibly important to actually understand, how is your compound binding? So is it protein-protein interaction? Is it a lipophilic center? So is it fat loving? Is it a hydrophobic center you're trying to probe? And really all of that guides the sort of series of compounds that you are to make. So that process, I'm saying, is at least a couple of months. So once you then identify the compound you want to make, you'll then do probably about a week of planning to see what chemistry is available on those. And then it will take maybe, again, a couple more months to synthesize those compounds. And then you'll put them into, you'll hand them over to a biologist or a biophysicist who will screen those compounds against the protein. And then, touch wood, you'd hope that they actually bind, but I'd say 90% of the time they don't, or they need further optimization. So you'll then see from those structures that you've made, which part is actually important to delivering the potency. And then you'll go back, you'll redesign, keeping in mind one of the functionalities in your molecule that you know is important. And then you'll go back and you'll build more functionality or more scaffolds around that core structure. And then you essentially just keep doing this process over and over again until you've got a really potent drug molecule. From there, you'll then give it to, you'll do some more toxicity issues on it. Again, more in vivo, so not in animal. Yeah. So you do like cell-based assays to see what the clearance is like. So for drugs, you don't really want a highly cleared drug. You want it to stay as long as possible and have a high therapeutic index. You don't want to take it and then 20 minutes later, you're excreting it via the urine, for example. Once you've then got a good toxicity profile, so a good balance between potency and mitigating toxicity, you'll then put them into an animal study. So here within the school, specifically for the DDU, we have rat and mice models that we can inject and essentially just analyze them. And then really depending on the sort of the potency and the drug profile, that animal study can range between you're dosing them once a day for 60 days, or you're dosing them twice a day for a week or so. It really depends on what your goals are for your project. And then hopefully, once all that is done, you then move into a clinical trial. But it takes years. So I have quickly summarized what takes years to do. I think there's some statistic out there from like identification of a hit to approved drug on the market is between 15 and 20 years. And it costs in excess of 100 millions to billions. Yeah. So it's a long process. And there are people within the DDU who have seen it from start to finish. There are different candidates. So, for example, the DDU malaria drug is one of the fastest drugs that have moved from its identification. So that is, you've identified the protein and you've identified the molecule, all the way to potency screening, toxicity issues, to phase 2 clinical trials. But that's more of a case because malaria is so deadly. Like you don't necessarily have 20 years to keep exploring when about 450,000 people die a year of it. So there's a battle between how important is this to get out and what is the profile of the drug. So that's wrapped up for chemistry. I don't know if biology, Connie, you have any more?
Yeah, I would just say that from a biologist perspective, if you are probing a system, if you want to find out if a protein is therapeutic or not, if you're able to establish that, then I guess the first thing, at least from an academic perspective, would be to do, and I don't think this is the same for industry, is to explore the protein pathway. From what I understand, industry tends to be much more, okay, we have a target, so now let's make a compound for that target. And if it's toxic, it's toxic. But if not, there's no interest to some extent about the greater question. But yeah, I mean, you follow through what Fergus basically said is once you have your protein target and you can validate, and the way you would validate as a biologist is you would probably do a recovery experiment in vitro. So you would do it within, or in cellular technically, but you would do, you'd start with in vitro for the thermodynamics and to validate that you have oh sorry, no, go the other way around. You'd start in cellular, and you would probe to see if you knock out your protein, does it induce the phenotype that you desire? And then once you've established that, you then start doing compound treatments, or you would do some sort of screening assay, which, you know, you would tend to, from what I understand, there's different ways of doing it, but from what I gather, the best ones to do tend to be fluorescence based. They're quite useful, especially if you're interested in protein-protein interaction. If, for instance, your compound engages with or disrupts a protein-protein interaction, you would do some sort of fluorescent assay, which involves the close proximity of two different fluorophores. That'd be like a FRAP assay or bioluminescent assay. And then, yeah, you'd follow on. You would just effectively, from the biology perspective, you'd ensure it's not toxic, and that's partly where you'd start using mouse models as well. Once you've established that it's not toxic within the cell, and I think there's the other element of that is the Lipinski's rule of 5. Isn't that one of the things to ensure that you can actually get your compound into a cell, and it's of a good nature, as to be a compound, to be a drug, and then you would do from cell, you go in vivo. Yeah, and you would just essentially do toxicity studies by observing mice, because mice obviously live faster than we do. It's a quicker model than giving it to humans, and then obviously we just go up the chain until it can be approved. And yeah, like you said, 20 years. It's a very expensive process, which is why a lot of drugs are very expensive. Yeah, I think the drop-off rate, or the sort of the failure rate for most drugs is somewhere above 90%. So yeah, I mean, if you're spending millions and millions and millions, or hundreds of millions of pounds on individual drugs and they all fail, you know, you have to recoup the cost somehow. So, but yeah, I think you just kind of follow it through that way. But yeah, I think at least from the perspective of a biologist, recovery experiments are fundamental. They're really useful to show that you're actually getting the effect you suspect you're getting. And that's the thing I'm planning on doing quite soon. So there's those kind of experiments. So
wow, that's amazing. So fingers crossed on those first experiments that you're doing then.
Hopefully.
Your recovery experiments. Yeah, so it sounds like it's quite a long process, really, getting from validating your initial target and designing a drug that's suitable to target that, or a series of drugs, sounds like, to getting it into the clinic is a very long process. Yeah, thank you for describing that. Perhaps we can move into some more general questions about the PhD program again.
Yep.
So both of you are clearly doing some great work on your research project. So I'm interested in what training you've had as part of your PhD programs. Connor, you're nodding. Perhaps you can start.
Yeah.
Thanks, Connor.
So in terms of training, so we have at the university, and I think most universities are the same in some sense, is that there is postgraduate training programs that are generalized to most postgraduate students. So it's not just research scientists or their ilk. I think it's also available to staff as well. So I think postdocs are able to do this as well. But these are sort of more general programs for instance, if you're an international student, I know that they run a quite successful writing program for writing in English, obviously, because if you're writing in your second language and it's the thesis, I can't imagine how much more difficult it could get. But yeah, they do things like that. They do Excel, and more particular to the MRC, in my experience, is that part of my grant funding comes with extendable or additional funding avenues for doing specialized training. So from what I gather, I'm able to apply and go for instance, if I'm interested in doing a CRISPR project, or if I want to learn microscopy, and the university doesn't have those facilities, I would go somewhere else and the MRC would pay for it. And also, I think there's an avenue in there for me to do some computational stuff, because my particular program is the QBiomed, which is technically quantitative and interdisciplinary biomedicine, which means I technically am supposed to be doing a project that is a combination of both computational and biological. But I have done no computational as yet. And yeah, I think in terms of more specific training, so I've been trained on how to do the FACS or flow cytometry, and well, yeah, sorry, I've been trained to do flow cytometry. I have to pay for FACS, but yeah, I mean, basically I've been trained on how to use, or at least work on one of the flow cytometers at university. I've been trained in microscopy, and these are things that you can do that regardless of your field. I think as long as you're willing to take the time out, you can get trained on these advanced technologies, which is fundamental if you're a biologist. And I even think, to some extent, I think the DDU allows me to go and do some of their assays, or at least I've been in that part of the building where there's the implication that I could get that training. So I think there's a lot of different ways you can get trained in either very specific or very general subjects at the university. But yeah, there's lots of ways you can learn, and these are some of the things I've taken advantage of in the past. So
great, thank you. Fergus, could you also discuss what training you've had?
Yeah, so luckily Connor and I are both funded by the same research council, so his opportunities are the same as mine. Obviously, being a chemist within School of Life Sciences, the training is more focused towards biology. I mean, I've done some of them just because I want to learn more about even the basics, like how do you screen an assay, like that is important for what I want to do, but I don't have to have the hands-on experience to know how to do it. I think for generic skill training, the university is great. I mean, so I think within your first year, or at least as part of your PhD, you have to do the core training programs, and it will get flagged up to your committee. So within the first year, I did a training program on how to read a paper and then take the important bits out of the paper, which when saying it like this, it sounds, well, obviously you know how to read a paper, you know what bits are important, but there is a skill to it. If you have about 20 papers that you need to read, once you've got the know-how of how to, right, I need to look at the title, read the abstract, ignore the introduction, because largely the introduction is just explaining why they've done the project or what they're looking at, and then go to the conclusion. And then you had to write a piece to a non-scientist about the paper that you've just written. So it's using two skills: how to communicate with a non-scientist, which in itself is a huge skill and incredibly difficult to do, and then how to read a paper properly. And then there's also training on thesis writing. So what sort of structure should you take for a thesis? Because at the end of the day, the thesis is a story. It's a factual story of what you did, why you did it, and what makes your story different from someone else who's doing a broadly similar thing. So it's all very useful. And then kind of at school level, we have these seminar series. So that's not necessarily training that you sign up for, but it's always good just to go along and hear what other researchers within your department or the school are doing, just so you can get a good overview of what's happening. And then for the MRC, we have a yearly MRC retreat, I think, is that right, Connor?
Yeah, once a year.
Where all the MRC students have to either present a poster, present a talk, or give a flash 5-minute talk. So that's a 2-day retreat. So you're sent off-site. You don't have to pay anything for it. So all your accommodation, your travel, your food, that is all paid for. And yeah, you can just mix with other MRC funded people. So it's good on essentially the making the friends front to it. Like, you know who else is in your funding body, as well as just know what sort of other disciplines or research the MRC funds. Say, for example, you want to go down the academia route and you want to move up to become a PI. It's always good to know what your funding bodies will actually fund. And then getting that from an early experience or an early age or early stage in your career is valuable, especially when it's free. So
yeah, that's great. So as well as having the training, which is quite specific to the techniques which you're using in the lab, you're also getting training on more general things which are useful throughout your PhD, like your writing skills. Do you also have training on statistics and data analysis and things like that? What kind of training do you have in that area, Fergus?
So statistics ones I haven't attended, mainly because it's not directly related to my discipline. So I don't know if you've attended it, Connor.
So I've attended. There are many more than the ones I have attended. That may be true. I have attended at least one. Yeah, no, there's definitely stats courses. In fact, not only that, part of our initial training when we start PhD is a 4-week training summer school, I guess you'd call it. A 4-week sort of extended sit down where we get basic training on most general aspects of biology, at least from a biology perspective. I think even a lot of the chemists do it. I'm not sure if you did, Fergus, but a lot of the starters in September go through this 1 month of just extended, and part of that comes with stat training. But yeah, I mean, every year they offer, I think, 3 or 4 different courses in stats, and R training, R programming, which is the bane of my existence. But yeah, R is something that the university likes to work with. And yeah, I think if you're really interested, there's also, there is funding available for all PhD students for the seismic course, which is sort of an advanced computational stats course that you can do that teaches you not only programming, but quantitative statistical analysis. I know this is fundamental. I think, like Fergus has said, I think this is more important for someone like me as a biologist, because obviously my N numbers tend to be more lower, so I need to be correct in my analysis of my research. But yeah, I think they are available, and they're plenty, because stats is so fundamentally important to doing any research. So.
Thank you. And in terms of ethics training as well, have you had training, for example, on research integrity or use of data?
Yeah, that's every year, from what I gather. We do, at least, I think there's at least 1 lecture a year I get on research integrity. But yeah, we get a lot of not only education on the subject, we also got a lot of guidance. There is an open door policy with a lot of the HR here at Dundee. So if you do feel like there is an issue with, an overly pressurizing PI, for example, which is one of the indicators for, the whole research ethics thing, because obviously if you're feeling very stressed, you could, in theory, start maybe not fabricating, but it's one of these sort of things you need to watch out for. So yeah, the HR department's there, and also we do get informed on, there is resources out there that we have access to sort of explore that subject, and we all know what is and what isn't considered scientific fraud. So
yeah, touching on from that, like one of the key things that they stress is like a bad result is a result. There's no point trying to lie about it or deceive it, because say for example you publish a result that isn't true. So I think there's a case, the Nobel winner for chemistry. She's done great, fantastic work over her career, but the one paper that she got her Nobel Prize for, they couldn't replicate it. And they found out it was an issue with the writing up policy. So they had to essentially, she still got the Nobel Prize, but they had to strip the paper from which she published it from, simply because someone tried to reproduce it and they couldn't. So yeah, it's kind of said as like, don't do scientific fraud, because when you do get found out, the repercussions, it's kind of like, you've got a mark on your back if you want to go into the career into academia. They'll know, for example, Fergus Preston lied about some results to get into a Nature paper, and you're essentially completely disbanded from Nature. You'll never get into that journal, but then subsequent journals will know about you. As for the ethics as well, as Colin said, it's a yearly course where you're kind of told about discrimination, science. Look, I mean, it's bad to say, people will probably subconsciously discriminate or have subconscious bias, and the course is kind of there to make you aware of it, aware of the bias that you may have, both scientifically to your results, to individuals, and just makes you aware and tries to teach you not to do it and be a good overall science ambassador. So it's important.
Thanks. Yeah, those are very important parts of your training to make sure that you're a good ambassador for science. And so most of your training happened within the university, but you have funding to access training outside the university as well if there's a more specialized course that you need to go on. Is that correct?
Yeah.
Yeah.
Okay, excellent. Moving on from training, I'm interested in what kind of social activities there are for PhD students, ways to interact with other people within life sciences, perhaps outside life sciences as well.
Yeah.
Yeah.
I mean, when you start a PhD within School of Life Sciences, you have what is called the Pickle Society. So it's the PhDs, postdocs and PIs School of Life Sciences Society. So they put on a range of activities. The monthly one, which most people are familiar with, is the Pizza Friday. So they order in hundreds of pounds worth of pizza and provide alcoholic beverages, soft beverages, free of charge. You have to do absolutely nothing. You all congregate in the school and you just sit on sofas and you can chat with either your friends or people you haven't met before. And then they have other societies, so like a Ciulli dancing society, singing, art. So they're all widely available, and they send out an email saying, "Hi, we're part of the Pickle Society. We're doing this. Would you be interested in attending?" And half the time, even if you're not interested in attending, it's just good to go to meet other people outside of your discipline. I think for once every year, they do a pickles retreat. So again, that is where you go off-site and you do like team building exercises, or they give you a range of sports or just things that you want to do. And again, that is all completely paid for by the school. So the school understand how important it is for people to interact, both for mental well-being, but also for scientific endeavours. But yeah, they paid for 2 nights accommodation or 1 night accommodation, all food, all travel, and all activities. So yeah, it is heavily funded by the school, and it's really quite nice.
That's great. And Connor, anything?
Yes. Well, I'd just say any of the more university general activities are also available to you. I mean, all the sports facilities are there, all the sports teams, and like Fergus said, the societies are there as well. So if you're interested in, I can't even tell you all of them, but there is anything from art to dancing, swing dancing, to some sort of appreciation. I'm pretty sure there's an environmental group as well. So there's a bunch of different out of research facility type society social gatherings. I think I made this point before, but the good thing about Dundee is it's quite a low cost of living city, which means that even a PhD student with a modest income should be fairly okay. And one of the benefits of that is that saves a lot of money for you to go out. And yeah, there's not short of any experiences in terms of going to pubs, going to restaurants. And Dundee's great for that. And I tend to find a lot of my social engagements actually come from my lab. We have at this point, mostly weekly / monthly get-togethers, where occasionally Fergus is also there. But yeah, I mean, and there's lots of stuff like pickles. Pickles is great. Free pizza on a Friday. Who doesn't love that? And yeah, and then there's also Creef, which is is an educational weekend that we do, but it's also a very social weekend. It's very famous for being a very social weekend. So yeah, it's but there's plenty of opportunities to socialize, and I think one of the benefits of the way the PhD programs are run is that when you start in September, you tend to make a lot of friend groups very, very rapidly, because of the way you're sort of structured into sort of small groups for training and for induction and stuff like that. So yeah, there's no shortage of opportunities to make friends. Definitely at Dundee. So
that's good. Yeah, it sounds like there are a lot of opportunities there for interacting with other people, both within life sciences and elsewhere in the university. You touched on Creef. Could you explain a bit more about that one, please?
Yeah, so Creef is our entire research facilities yearly conference, where the research that has undergone over the year before is sort of presented to everybody who's interested in going, which tends to be quite a large percentage of the total number of people at the facility. I think it's like something like 40 or 60% total. Everybody goes, and it's essentially it occurs at a hotel, the Creef Hotel conventionally, but it's done over 3 days, where it's a whole day of lectures. But then at the end of the day, we do social events. We have nice dinners, and yeah, there's quite a few different pubs and various other activities available, bars available for drinking, and it usually ends up being a really, really enjoyable experience, especially for young PhD students, because we don't have to pay for it. The first 2 years of our PhD is paid for by the university for going to Creef, and it's also a really good place in terms of exposure. If you want to either present work as a PhD student in your 3rd year, and you actually have the nerves to do so, which is something I don't have, but if you're able to stand up in front of, I don't know, Dario Alessio, Philip Cohen, and all these bigwigs and present your work, nerve-wracking but awesome. And also, it's good just to meet other PIs in a non-academic setting, where they're maybe not quite as sharp as they normally are, because they've usually had a few drinks. It's usually a great experience, and you tend to meet interesting people. It's a very interesting experience. I really enjoy Creef. Pity that we had to skip it this year due to COVID, but hey ho.
That's great. Thanks for explaining what Creef is. Yeah, and certainly it's a good place to, when you're first putting together your research story, and maybe you've got a paper that you're trying to write, it's nice to get some feedback from all the other researchers in life sciences. It's a bit of a protected environment to get some feedback on your research story.
Yeah.
Okay, that's great. I have a question. I don't know whether either of you have had to change your research project during the course of your PhD, or if you know of anyone who has and can comment on how that went.
Fergus?
I mean, for me, I spent the first kind of 8 months of my thesis, my PhD, trying to synthesize just one compound, because I thought it was promising. The computational said it was interesting, and then it got to the end of the 8 months, and I essentially just had to bin it. There was just no point. There's no point carrying on with something that was just a dead end. Like I could have made my whole PhD about how I synthesized and overcame and developed new methodologies to synthesize the compound. But when you were to almost relate that to my title, which is the synthesis of fragments, plural, there's no point wasting months, years on one thing. I mean, it's not necessarily a change, but I then had to go back to the drawing board again, look at my fragments of interest, and then pick a couple, and then do all the literature searching and then the chemistry on those. It can be hard to give up because you are so passionate, you know, so much about that one thing you're doing. And for me, yeah, it was disappointing to give up, but when I had the conversation with my supervisor, we kind of both knew that we had to move on because the project has to progress. And as much as you know that you have to do it, doesn't make it necessarily any easier to give up. So I almost thought it was a bit of a failure. I hadn't been able to synthesize it, but the supervisor was like really good about it. He was just, look, this is a PhD, things aren't meant to work. You're investigating something completely novel and new. Like, if it was easy, everyone would do it, that famous saying. And it's true. Like, as soon as you realize that moving on isn't necessarily a failure, it's just a progression of your training, because that is essentially what PhD is. It's scientific training that you then get a qualification out of. It's just, yeah, it's an invaluable lesson. Yeah, I'm not going to sit here and say a PhD is easy because it's not, and you will have these obstacles that you overcome, be it a change in direction, something doesn't work at the last step, or even like supervisor issues. You might have a disagreement with your supervisor. So yeah, it's difficult.
A PhD can be challenging, and certainly things don't always go in the right direction. They don't always work.
Nope.
And certainly for my PhD, I had to change direction after about 8-9 months. And yeah, but you still, during that initial period where you're working on your first project that didn't quite work out, you still picked up a lot of techniques and a lot of made a lot of connections with people in other labs as well who can help as you move to a new project. They can be providing reagents, providing ideas, or just providing support, which is really beneficial. So, yeah, I'm glad you have moved through that. It's like difficult step of changing direction. Yeah. Okay. And there is a comment from someone who's attending who says, "Never give up and good luck." So that's a great message for you.
Thank you. Yeah.
Okay, that's great. I'm just going to check that we haven't missed any major questions here. Oh, there's a question in your project, Fergus. You've got an industrial collaborator. The question that always comes in when someone says they've got an industrial collaborator: are you able to publish your data? Do you have an agreement in place there?
So that agreement kind of goes above what I know. I'm just speaking with my supervisor and industry. I know that there is an agreement there. So it really differs between complete transparency, so whatever you publish, they're happy with, and vice versa. But say, for example, if I wanted to use one of the techniques that my industry has developed but have not published, they might not necessarily want that disclosed. So that goes from all the way from publication in the paper to even discussion in my final viva. So I'd either omit it completely from the thesis, or if it is in the thesis, you can apply, I can't remember what it's called, but it doesn't get published in the end. There's some sort of clause that you say, no, you have to wait so many years before you can. I mean, it's quite nice because if there is that, your committee member can't ask you a question about it because it is completely confidential. But yeah, the law and the agreement, I don't really know much about it, but yeah, there is definitely transparency and confidentiality agreement that we do have.
There's usually fairly specific between the researcher and their industrial partner, so it varies a bit case to case, I guess.
Yeah.
Okay, thank you. And there's a question in asking, in terms of changing your project direction slightly, you initially are submitting a title for your PhD. So is there a way to change that partway through if your project changes direction?
Yes, I think so. As long as you're not deviating so much away from essentially what you got funded to do. So whilst my title is pretty broad, development of fragments for fragment-based drug discovery. That is my title that I saw on the application. I mean, I can take any route from that I want. I can say the identification of these sort of compounds for fragment-based drug discovery. So yeah, you can deviate. The title isn't set. the theme of your project is set. But yeah, the type you can deviate, but I'd say deviate slightly away from what was given to you. I don't know if you've had any experience with that, Connor.
So yeah, I mean, like, maybe not to be cynical, but the way you kind of structure your PhD application, you kind of make it as broad as possible for that particular reason. And in my experience, what I did, my project title, although I'm more specific in my presentation, I think the actual one I have is the development of novel degron technology to explore protein biology, which means I can go anywhere with it. So, and for that reason, it's just so that I can, yeah, I mean, for one thing, like Fergus said, he's had some failures. A lot of my successes have basically been on the backs of almost 1.5 years of complete failure. So, I mean, yeah, it's part of the process. And I've had to change my sort of, you know, I had this goal, now I'm sort of shifted to this goal, and now I'm shifting to this goal. And just kind of the way I've sort of done that, it's just so that you can get a coherent story in the end. And yeah, I think it's, yeah, I think although you have initial title, I don't think they would even falter you that much if you have to deviate to some extent. I think everybody would deviate to some extent. So I don't think anybody who goes into a PhD already knows the outcome. I don't think it would be a very good PhD if that was the case. So, yeah.
Thank you, Connor. So we're also joined today by Professor Kim Dale. Perhaps, firstly, thank you for joining us, and perhaps you could introduce your role within the university.
Yes, absolutely. Thanks, Nicola. So my name's Kim Dale and I wear a number of hats at the University of Dundee. So I'm a researcher. I have a group in the Division of Cell and Developmental Biology. I'm also the Associate Dean International for the school. So I oversee the development of all sort of international partnerships, collaborations, whether that's at undergraduate, masters, or at PhD level. So I'm absolutely delighted that we've been able to engage and participate in today's webinar, and our fantastic PhD students acting as ambassadors and giving their views on what it's like to study here. So absolutely delighted to be here, and yes, if there's any questions, I'd be happy to field those.
Thank you, Kim. Perhaps, so a few questions have already come in, and perhaps I could start with this. So someone who's looking to find a collaborator for PhD research, are you able to advise them how to go about this?
So would this be for a student looking for a potential PhD post? Is that?
I'm afraid I don't know.
Okay, so I'll interpret it in that way, but please do email me if I've slightly interpreted that wrongly. So I guess if you are, you know, very keen to be looking to the next step and to undertake a PhD, and Dundee as a destination is something you're thinking about, and you're trying to work out sort of what area, what research discipline or field to pursue, then the best place to look first is our school website, where our different research divisions are clearly outlined, and then within each of those divisional web pages there's a list of all the individual group leaders and their web pages that talks a little bit about the research going on in their labs. Another route would be to go to our PhD web page, where the different PhD programs are outlined, and what the differences and the details are within each of those programs, and again lists of projects that have already been put forward by the PIs. So that's normally the way that it works in life sciences at the University of Dundee. That is, that the group leader puts forward a project to which prospective applicants then apply. Within that, there is of course scope for you to contribute your ideas in the way and the direction that the project goes, but initially you would need to apply through that route, identifying which project or projects you were most interested in.
That's great, thank you, Kim. I think that asked the question about how to find out a little bit more about the PhD programs and the research at Dundee, as well as how to apply, which is of course signposted on the website. We have some questions about the research commercialization process. Could you talk a little bit about that and how it works in the School of Life Sciences?
Yes, delighted to do that, Nicola. So obviously, you know, science is ever moving and ever evolving. And this idea of linking, you know, basic research discovery science to commercialization and translation is something that Dundee is really committed to, and it's one of our school strategies going forward. We actually have a commercialization and translational committee that look to support labs that perhaps have got a project that has the potential to develop either into a spin out or linking with different industrial partners to commercialize their ideas. And so there's a specific committee there ready and whose remit is to support both the PI, the postdoc or the PhD student in how to take that forward and how to develop it. So, you know, I think it's hard to come from the very sort of, you know, first step of deciding that you want to do something in the commercialization translational direction. What normally happens is you apply to a project, and if that project then takes a turn that allows it to go down the commercialization or translation route, then that's when this committee would come in and support you. Of course, it may be that you join a lab that's already on that journey, so they already have projects that are geared towards spin-outs or commercialization or translation, or in, you know, strong industrial partners that they're already involved in. And so that may well be outlined in some of those PhD projects that are listed on the website. And if that is something you're specifically interested in, it's worth perhaps looking at projects that have that already embedded within them. But if there's a project there that you're really keen on that doesn't yet have that link, it's not to say that that wouldn't develop in the course of your project. So again, if you have a project that you've identified on the website you're really keen to pursue, it might be worth contacting that PI to sort of say, is there a view or an ambition to develop this into something that might go along a commercialization or translation route?
That's great, thank you. So there is a specialised committee that will help support as you try and transition towards a startup or commercialising your idea. There's a question, is there experience of working with other countries to implement these commercialization practices?
Yes, I mean, I think, you know, we're driven by the science. So the strategy is that, you know, you identify the partners in whatever country that may be that's going to take the science forward in the best, you know, most effective way. So there are no global boundaries in that regard. So if anything, you know, developing networks with international partners and institutions is something that we're very strongly committed to, and something that falls very much well within my remit. So I would be absolutely delighted if we could increase our footfall and our global reach in developing those commercialization routes. Yes.
That's great. Thank you very much, Kim. I think that we've covered most of the questions which have come through now. So perhaps we'll go to each of the panelists to hear their final thoughts before we end this session. Kim, would you like to start us off, please, on any final thoughts or messages you'd like to share?
Yeah, thanks, Nicola. So I guess, you know, what would my message be to you? I guess, you know, my personal message to each one of you would be that embarking on a master's or a PhD course is one of the most challenging, but one of the most fulfilling and exciting adventures you will undergo. So, you know, if you're a little bit nervous or a bit cautious or a bit worried if you're going to be, you know, able to go through with it, don't worry about that. It's good you feel nervous, right? But you want to channel that into sort of motivation. And so I strongly encourage you to follow that dream. Obviously, I'm a little bit biased, but I have to say that I think Dundee as a destination is an absolutely fantastic option for you to consider. Firstly, from the point of view of the world-class research that we have going on in life sciences, you know, it's not for nothing that we are ranked as #1 in the entire UK for research in life sciences, both within the impact the environment and the output of life sciences. So, you know, above Oxford, above Cambridge. So if you're thinking of a destination, this is a fantastic place to think about. And I'm sure myself and all of our wonderful panelists here would be absolutely delighted to hear from you if you had further questions on any of the topics that have been or haven't been covered today. But yes, so my word of wisdom there is to just follow your dream, and you know, you can do it if you really want to.
Thank you, Kim. That's a really nice message. Fergus, could I have your final thoughts, please?
Yeah, kind of just echoing Kim's. The PhD Masters is a journey. It will be difficult. It will be challenging. You know, I felt like I've spoken quite freely about my experience of it. My advice: only really embark on a PhD if you're truly passionate about your discipline. If you don't necessarily enjoy it that much, you will find it 10 times harder than someone who is fundamentally passionate about what they're doing. And then, as Connor said, and Kim as well, we are the best school of life sciences for research above Oxford, and our living expenses is cheaper. So both for your academic and for financial reasons, Dundee is just this fantastic place to be within research. And we are becoming the city of discovery. Money is being pumped into Dundee, and yeah, it is evolving at quite a fast pace, and it's just a really scenic place to be. I enjoy it.
That's great. Thank you, Fergus. Connor, could I have your final message, please?
Yeah, I mean, at this point, it's effectively echoing the two previous speakers. But what I would say is, going into my PhD, I was very apprehensive about starting. I guess because it's from the outside, it seems almost impossible to think that you can write 50,000 words and take 4 years to do so, and do all that sort of stuff. But what I would say is that what I've found is not only in particular maybe Dundee, but I would say in general, the PhD has been I've had a lot of guidance, a lot of support, and fundamentally I've really enjoyed doing my PhD up to this point. I don't know if that will continue in the future, but I can say for the moment I've really enjoyed doing this PhD, and I think for anybody who has had any lab experience and enjoys this aspect of science, I would implore them to try a PhD, and I think they won't regret it. I certainly haven't. And yeah, Dundee's a great location. Not only is it cheap, which I can't stress enough, because you know, if we were in London, there'd be a completely different story. But also great connections. Dundee is a lovely city with lovely people in it. I've lived here almost all my life. I love it to bits, and yeah, and we have a very international community as well, and you know, it's everybody here wants to be here, and it's just a really great environment. So yeah, I mean, Dundee's a great place to do a PhD, and yeah, doing a PhD is also very good. So yeah.
Great, thank you, Connor. So just to highlight, our panelists did presentations to outline their research, recorded those, and those are available on the Kazakhstan PhD Association website. Links to those are also available on the Facebook site as well. And there was also a discussion from life sciences researchers during the previous webinar, the drug discovery webinar that was part of this series. So you can also check that one out as well. So thank you to all of our panelists for the help with this discussion today, and I wish you all the best for continuing your research, and to Fergus and Connor for completing your PhDs. Thank you to the Kazakhstan PhD Association in the UK for your help with organising this session, and thank you to everyone who's attended and has submitted their questions for helping to contribute and guide this discussion. So the next webinar in this series will be tomorrow at the same time, and that's focused on energy, mining, oil and gas. Thank you, and that's it from us here today. Goodbye.
Thank you.
Bye. Thank you.
Bye, Nicola.